Well, all right, everyone, we can get started. So before we get started, I'll just mention after the seminar, we'll be going across the street to the Mexico window for drinks and appetizers. If you'd like to join. Thanks for coming. This is Seth Redman. He's a professor at Washington State University. He does a lot of really cool work looking at eco-evolutionary dynamics across a number of systems, ranging from fish to flies. The sort of running theme in his work is evolution on relatively short timescales. Before starting at Washington State, he was a postdoc with Paul Schmidt at UPenn, and prior to that, he worked with Dolph Schluter at UBC.
Thanks, Mark, for the introduction and for the invitation. It's been a lot of fun to come down. I'm at WCU Vancouver, so not very far up the road. So if you see anything here you want to chat about potential collaborations, I'm a new professor just like Mark, so looking for more local contacts, more people interested in talking about evolution and its ecological consequences. So, I like to start with sort of a broad sense of motivation, and what gets me interested and excited about biology is biodiversity itself. I mean, the fantastic, exciting biodiversity like chameleons, but also intraspecific genetic diversity, things like stickleback, variation in phenology in trees. And over time, I've moved from the more exotic to the more pedestrian as I've tried to study mechanisms that underlie changes in maintenance of genetic diversity. And so Mark mentioned that I'm interested in evolution, rapid evolution. And so what do I mean by that? If you think evolution in a Darwinian sense, you think of sandstone cliffs slowly and imperceptibly shaped over eons, this very slow process. It's studied with fossils, it's studied retrospectively only. That's probably not the view of most folks here. You've got Mark, you've got Molly, you're probably not thinking like that. The other end of the spectrum, I like this quote from Dick Lewontin, who is an eminent evolutionary biologist, who says, "At every moment, natural selection is operating to change the genetic composition of populations in response to the momentary environment. But as that composition changes, it forces a concomitant change in the environment itself. Thus, organisms and environments are both causes and effects in a co-evolutionary process." I think this is kind of a magical perspective to think about ecological and evolutionary change occurring at the same time. And it sort of presupposes this field that's emerged largely over the past 20 years. It's called Eco-Evolutionary Dynamics, that investigates these reciprocal interactions. But at the time this quote was made, it like way outstripped our understanding and our data of how ecological and evolutionary change interacts, at least outside of just a few systems. So when I think about this question, there's sort of some sub-questions within it that I study often in my research group. And the first is, what is the time scale, pace, and magnitude of adaptation? So if we're talking rapid, how rapid are we talking about? But not just how fast, how much. So how much trait change, how great of a change in allele frequency over what unit of time? And genomic data really help us to do that, because without genomic data to measure evolution, you have to pick some subset of traits. You hope they're ecologically and evolutionarily important, and you're limited by our very limited capacity to measure evolution in them. So it's somewhat easy to measure evolution in body size, but to measure evolution in some physiologically important process could be really challenging. Genomic data give us an easy out, or another source of data with which to study evolution in much higher resolution. So oftentimes do that. But genomic data have limitations. They only really allow us to infer patterns, especially if we're studying them after the changes have occurred. So if you study population A and B, they've been diverging for some long period of time. You don't know much about their demographic history. It's hard to ascribe process to pattern once you're pretty far removed from the action of those processes specifically. So it doesn't really help us much understand the process of adaptation. So the second question is, does rapid evolution alter ecological and eco-evolutionary outcomes? Things like species interactions, population persistence, and sort of dynamic feedbacks between ecology and evolution. That's sort of at the heart of that Lewontin quote. That's really important for things like population persistence in the case of evolutionary rescue. And I don't think we have a great sense for how important rapid evolution is in ecological outcomes in systems in which we know quite a bit, even about their ecology and evolution. We don't know how much those things actually interact. And so what I'm saying that And the way in which evolution can impact ecology is you could have interest-specific variation within species that shapes ecological interactions. Think about evolution in beak depth could shape the interaction with some things that a bird feeds on. You could also have local adaptation itself, an overall change in a number of traits influencing vital rates from a population perspective, the rate of recruitment and population growth. So the third question I sort of think about is we have rapid evolution, what drives it, how rapid is evolution, does it alter ecological dynamics? And then the third part is, are there actual reciprocal feedbacks? And that Lewontin quote, he's talking about this dynamic process where ecological changes occurring, it's driving evolution, the environment, or the evolution itself is driving changes in the environment, there's this dynamic system. Are these feedbacks between these processes, are they real? Are they realistic? Are they important for either ecological or evolutionary outcomes? These are some of the bigger questions that I think about in my work. So let me convince you why I think these questions matter. I think they intersect with a couple of different areas in evolution and ecology. So why do I think these questions matter from a population genetic perspective? Population genetics, studying rapid evolution helps us answer some questions about whether genetic variation is overwhelmingly neutral, which has been a debate for a number of years and sometimes feels like it's getting resolved, sometimes feels like it's just getting more vocal. And then not only that, but slow molecular evolution is really central to a lot of population genetic models. And can we test and refine some of those models with empirical data? I really like this paper by Messer et al. About population genetics and what it can provide and what it says in cases of rapid evolution. From a quantitative genetic perspective, there's been a lot of focus on major effect loci, large effect loci with very easy to observe phenotypic effects like this variation in bony armor plating and stickleback that I worked on and still work on some. But what is the genetic and phenotypic basis of adaptation in natural populations? In contrast to this variation that we see that's discrete and highly studied in a number of systems, when we look at the genetic basis of trait variation and things like human height or other complex traits, we find that there's so many genes involved that it becomes almost omnigenic. It seems like almost every gene or some huge percentage of genes is involved in dictating the phenotype of some complex traits. And so how do we square these things together and which sort of between this continuum or these two extremes between the single gene having a huge effect on phenotype and many, many, many genes having an effect on phenotype, how does that help us understand or predict the outcomes of evolution? And from an evolutionary ecology perspective, I think these questions are important because we don't know how much of a driver rapid evolution is in ecological outcomes. We don't know whether changes in genotype frequency are key to persistence of populations that are out there where we can't see evolution occurring necessarily very easily. And then also the idea that there's this dynamic interplay, that there's an interaction between ecology and evolution, and that's important for the outcome of both ecological and evolutionary processes. So that's why I think these questions are worth asking. And I've got a double slide, sorry. So the first part I want to address is, does rapid evolution occur? And if so, what drives it? I'm not the first person to ask this question. I'm not the first person to ask this question, even in outdoor experiments. There have been a number of people like David Resnick and Tom Shaner and my PhD supervisor who have built or constructed or done really awesome field experiments to measure evolution. So I want to pay homage to that. And one thing that I was doing when I was setting up these experiments is I was trying to think about agents of selection, things that may drive evolution that haven't been very well studied, things that we maybe didn't appreciate their importance in shaping the fitness or differences in fitness between individuals. And so one project I worked on was looking at the role of microbiomes as an agent of natural selection. And the reason why I think this is interesting is because microbiomes are interesting, and they're sort of newly interesting as our tools to study them have come of age. So with barcoding genes and metagenomic sequencing, we're pretty well able to characterize microbiomes and hence potentially study their effects on fitness and their ability to drive evolution. And so what do we know about microbiomes and variation of microbiomes in terms of driving differences in physiology or fitness of their hosts? Studying Drosophila here, what we knew or what we already did know before I started this work was that differences in association between an individual Drosophila genotype and different bacteria in the gut have big effects on key aspects of life history, like growth rate, digestion, survival, lifespan. Aspects of the microbiome are really key in dictating key aspects of host physiology. We also knew that microbiomes vary over space across populations. So in addition to being really important in shaping host physiology, they vary quite a bit. So this is the east coast of the US, you can kind of see that's like the Cape right there near Boston, and you're going down towards New Jersey and Philadelphia. From previous work, we knew that there's variation in the relative abundance of the microbiome. So in this figure here, the red bacteria is acetic acid, the blue bacteria is lactic acid bacteria. You can see there's some variation in the relative abundance of those groups in the guts of flies as you move down across latitudes. That's not unique to flies. There's lots of spatial variation in microbiome composition, which is somewhat hard to understand. Microbiomes have a huge effect on host fitness, but they also tend to vary a lot across space and time. So when I was in my postdoc, as Mark mentioned, I was working with this guy, Paul Schmidt, great advisor at University of Pennsylvania, and we were working in this outdoor system here, which I really enjoyed and I've continued to do work in. And so we built this orchard. And what it looks like is these are about 2-meter cubed mesh cages, and there's a tree inside, and it's trying to mimic the natural environment of Drosophila melanogaster in this case. So something similar to a fruit orchard is what we're trying to mimic here. And we use that for this experiment and several subsequent ones that I'm going to talk about. So in this particular experiment, we were looking at whether and how differences in microbiome composition drive evolution of Drosophila melanogaster. And so we had 3 treatments. We had an Acetobacter addition, so that's where we added Acetobacter to the food. A Lactobacillus addition, we had a Lactobacillus to the food. And we had a no addition, a sort of a microbial control treatment. These are just populations evolving in response to whatever natural microbes are on the food. And so the first thing that we weren't even sure we'd be able to achieve is we're going to be able to shift the composition of the microbiome. So microbiomes are controlled by many factors. They're controlled by host genotype, they're controlled by microbe interactions. So we weren't even sure if we'd be able to shift them in one direction or the other. But thankfully, when we added Acetobacter to the food, we got a higher relative abundance of Acetobacter. And Lactobacillus to the food, we got a higher abundance of Lactobacillus. And the overall community composition shifted a bit. So our treatment was an actual treatment. We were able to achieve some shift in microbiome community composition, but this isn't a wholesale shift. It's not like we mono-associated these flies with one or the other. We're just sort of nudging the microbiomes of these populations towards Lactobacillus or towards Acetobacter. So then we were studying some basic parts of the ecology of this experiment. So we were taking census size data. And this is when we first started to notice that maybe something was happening here. And so LB is Lactobacillus addition, AT is Acetobacter addition. And over a window of time, there got to be really big differences in density. So something was happening, interaction between the microbiome treatments and something that was going on in the experiment was leading to actual differences in the density of these populations. And so that got us excited that maybe we were studying an important dynamic here. So what did we do to study evolution and to determine whether these differences in microbiome composition actually drove evolution? Was we did pool sequencing of individuals from each of the experimental replicates, and we did it to high effective coverage. So we tried to be able to detect changes in allele frequency that weren't massive. So to be able to have a relatively good ability to detect these changes in allele frequency. And so what did we do? We sequenced those populations, and we also cross-referenced those populations, the changes in allele frequencies that we saw, with changes in allele frequency you see over space. So I mentioned earlier that microbiome community composition varies across latitude. Previous work had also sequenced these same populations across latitude. So we had a number of sites in which we knew the allele frequency, the shift from an A to a T or a G to a C at an individual site, was associated with changes in latitude, so clinally varying sites. And so we used those as candidates that were likely involved in adaptation. But the first thing that we did is we just looked over the whole genome at windows of differentiation between the AT and LB cages, and whether they were also enriched in variation in natural populations. And so here, this is a Manhattan plot. So I'm showing differentiation between cages across regions of the genome, each of the Drosophila chromosomes. And these, as it goes up, it's a smaller p-value in terms of larger differentiation. So this is showing the genomic architecture of differentiation between the treatments that we had. And you can see that there's a kind of a scattering here. There's a few different sites above a false discovery rate that show that we are seeing divergent natural selection associated with our treatments. Most of these sites are not sites that were involved in adaptation across seasonal or clonal time that were found in previous studies, but some of them are. So these ones that are not red were previously detected in other studies studying adaptation across space or time. So when we dug more deeply into some of these sites that vary over space, which is where we also see microbiomes varying over space across latitude, we tested whether the change in allele frequency in terms of the change from the major to the minor allele was concordant with the change in allele frequency we see over space. This is kind of a bit of a strange analysis, but essentially take it this way. If you think about an allele that varies over space where it's higher frequency of A here in the north and higher frequency of, let's say, C further as you go further south, that's also where you see a higher percentage of lactic acid bacteria and acetic acid bacteria. And so we were asking in our experiment where we either enriched acetic acid bacteria or lactic acid bacteria, did we see the change in the same direction in terms of a change in the direction of allele frequency that we saw over space within our individual experiment? And this heat map shows that we did. So the sites that showed the strongest divergence between our AT and LB cages also showed the strongest change over clinal space. And so something about our experiment in this individual agent of selection, this microbiome change that we saw, was recapitulating some of the changes in terms of the direction of allele frequency change we see in nature. So what do I take from this? Microbiome composition is a sufficiently strong agent of selection to drive rapid evolution. That was something we were interested in. I'm not wholesale surprised by that. This second part was really surprising, and it's still hard for me to wrap my head around it. That variation in microbiome composition seems capable of driving patterns of genetic variation across natural populations. We're seeing the same sorts of changes in allele frequency in terms of the same direction of allele frequency change that we saw in nature. So that particular paper, that particular story was about investigating how a specific agent of selection influences evolution. And what we saw was evolution is really fast, and that a single manipulation, in this case of the microbiome composition, was able to drive rapid evolution, and we were able to detect it genome-wide, and it showed some parallels with what we see in natural populations. But you don't need to create divergence. You don't need to drive changes in the ecology to drive evolution. If you're doing an experiment outside, there already are changes. There's temporal changes from fall to winter and summer, and there's just changes that occur naturally. And so we wanted to investigate some of that as well. So in this Lewontin quote, he's talking about the natural selection is operating to change genetic compositions in response to the momentary environment, in response to just the environmental change from today to tomorrow to next week to next month. And we were interested to the extent that which that was true, in which the extent that populations adaptively track environmental change in natural contexts. And so we used that same cage setup to do a relatively similar, but actually quite a bit simpler experiment. And what we did is we used these cages and we founded them with genetically diverse population. We put them into these cages and we tracked their evolution over time. We tracked it in two ways. We tracked it by doing the pool-based sequencing I mentioned earlier, where we sequenced them to a high effective coverage using a haplotype inference. We also tracked their evolution by measuring phenotypic evolution. So by repeatedly bringing these flies from the field back into the lab, rearing them in a common garden for a couple generations, and assaying some key phenotypes we think matter for the fitness of these individuals. What do we find? So this is the phenotypic data first. What I'm showing here is a set of 6 different phenotypes. Each individual gray line is an individual population, and this is over time. The red line is the population mean. So the first thing that strikes me when I look back at this figure is that we see evolution basically in all the phenotypes. Some of them, the strongest evolution is just from when they were founded, so released in the field to the first time point. That suggests just really rapid evolution from the lab to the field. But some of them, we see evolution that shows evidence of fluctuating natural selection. So where they initially were evolving to become more desiccation tolerant and then less desiccation tolerant. So something about the environment was driving evolution in one direction, and then the environment changed and drove it back the other direction. And this kinds of fluctuating selection was something we were sort of surprised to see, especially to see it that cleanly, like we see in desiccation tolerance and somewhat in development rate. So overall, the evolution that we saw here was really fast. Mark mentioned I'm interested in the pace. And one way that you can scale it is by looking at the changes in the number of standard deviations per generation. We call that a Haldane. And so this is a previous meta-analysis that sort of scored the rates of evolution of many things that have been measured, and basically everything been measured. And our red points here represent the changes that we saw in this study. Not just that our red points here are a bit more strict though, because we reared these in a common garden for 2 generations and assayed the actual change, the actual evolution. In these other studies, many of them were just measured from field collections without common garden rearing. So they included plasticity as well. So the evolution we see here is really fast. We didn't just measure phenotypic evolution, we also looked for change in allele frequencies over time. So we sequenced pools of individuals over each window of time. So that's like June to July, July to August, August, September, September, October. And so each of these, I showed a Manhattan plot before, which is looking at windows of differentiation. Previously, it was between the 2 different microbiome treatments. In this case, this is over time. So this is looking at the change in allele frequencies over a 1-month interval. So this is the first month of the experiment, the second, the third, the fourth, and this is the whole experiment, all of it put together. There's a lot of data on this, and there's a lot to make sense of here. But what I'll say, what I mostly think about when I look at this figure is the number of sites in the genome that are involved in adaptation over individual windows of time, especially the later 2. There's a lot of the genome is involved in adaptation to whatever conditions we saw changing over those windows of time. Also, we did some analysis looking at the direction of allele frequency change over subsequent windows. And so we did an analysis that we sort of colloquially called the leave one out analysis, and we were looking at the performance and the changes in allele frequencies in subsets of cages. Really, when you're trying to study adaptation, that's one of the strongest cases or the strongest lines of evidence you can have is parallelism. If you see independent populations changing in parallel, that's indication of a deterministic process. And in evolution, we're usually talking about, in this case, at least, natural selection and adaptation. And so what we saw is what we tested for were cases where we did see parallelism. So we quantified parallel change across a subset of 9 cages, and we double checked with the 10th cage that we actually see parallelism there, parallelism across all 10 cages. We looked at shifts, the windows where we see the most consistent and significant shifts over each window of the experiment. We see parallelism across each window, maybe least strong here at the end, this time 4 to time 5, October, November time period. But we looked at the sites that are showing parallel genetic change over an individual window. What were they doing at other points in the experiment? So these points here, these windows of the genome, they're the points that underlie this part of the figure are critical for evolution from July to August. But what do they do from August to September, September to October? And sometimes what we see is that points that are involved in adaptation over one period here are actually moving in the other direction the subsequent period of time. So selection's driving change from one allele to the other, and then driving it back in the other direction. So the same way that we saw fluctuating selection pushing in one direction and the other with desiccation tolerance, we saw that in the same way in many sites in the genome. So what did we take from all this? This experiment is really just a way of measuring evolution over time as natural environmental variation occurred. There really wasn't a treatment or control. It's just one temporal study of 10 populations. But what do we see? We see evidence that populations adapt in response to continuous environmental shifts, this continuous evolutionary change occurring. Okay, that wasn't so shocking to me. Parallel evolution is driven by strong selection on multiple phenotypes and genetic variants. I thought this was cool. So we measured a number of phenotypes. We measured changes in allele frequency across the genome. It wasn't just one phenotype under selection at one point in time, or 2 phenotypes. It was basically all of them under selection. The identity of the phenotypes and the variants under selection changes over a short time scale. So the direction changes. Which phenotypes are under selection most strongly changes is over pretty short windows of time. And selection can also operate at multiple time scales. So we saw evolution over each window, but also over the course of the whole study. I think the reason why the results of this study matter is that I would argue that rates of evolution have been systemically underestimated as they don't account for fluctuations. So when we measure evolutionary rates, we take a time beginning and a time end, and we look at the change. But during that time, our assumption is that the change has been linear when we estimate that rate. But if there's fluctuations in the direction of natural selection, this point from here to here may obscure all these changes in between. And so the momentary rate, the instantaneous rate of natural selection may be far, far higher than what we estimate with most data that we have. Another big point is that standard genetic variation is abundant. So the genetic variation in our populations was abundant, and many variants had effects on fitness. It wasn't just a few variants having effects. We saw selection over many, many different sites of the genome. It's hard to differentiate it from draft or from the effects of linkage combined with selection, but a lot of the genome was involved in adaptation, either due to selection or a combination of selection and linkage. So I think the takeaway is that adaptation may be an important process shaping ecological outcomes across timescales. So last question, I guess the second to last question about kind of combined, question 2 and 3. When rapid evolution happens, does it alter populations, communities, and ecosystem functions? I mentioned 2 mechanisms by which this could happen. You could have trait change leading to changes in communities and ecosystem structures, and I'll just talk about that very briefly. So this system of stickleback, which I don't work on as much anymore, but is near and dear to my heart, it's been well studied in part because of speciation that's occurred repeatedly between benthic and limnetic forms. This limnetic form feeds in the open water, primarily on zooplankton. The red benthic form feeds in the near shore, primarily on aquatic larval invertebrates. And there are cases in which, or at least one case in which, this divergence between this benthic and limnetic form underwent a reversal. So these 2 distinct, phenotypically, genetically distinct forms merged into one continuous distribution of traits and largely continuous distribution of alleles. And so this case of rapid evolution due to speciation reversal is one that I studied, looking at how this may cause ecological consequences. So this is a very rapid case of trait change where you have this bimodal distribution switching to a unimodal distribution over a window of about 6 years. So in my PhD, I asked questions about what were the consequences of this change. So the shift from this bimodal distribution to unimodal, how did that impact the benthic invertebrate abundance of the system? Or how did that impact the decomposition rate of the system? And as you might predict, changes in these functional traits due to this rapid evolution had widespread effects on the aquatic ecosystems in which these fish live. But the second part, and the part that I'm more interested in now, and perhaps is a little more general, is talking about what role rapid evolution plays in altering population growth rates. If you think about the question of being how many individuals in our population, where and do they persist, this is a situation in which I think evolution can have big effects. So let's sort of think about this as a thought experiment. So how might local adaptation influence population growth rates? If you imagine that this red Drosophila is a locally adapted individual, as the frequency of that locally adapted individual or genotype increases, you might expect that you see an increase in the overall numbers of individuals in the population. This might not always be true, but perhaps this is a general expectation. So does rapid evolution alter population persistence and dynamics? And if so, can we test for a feedback? So we did more Drosophila experiments. We didn't just build this orchard for the 1 or the 2, we did another one in which we, the first part of it, the control was the experiment I talked about previously. We just put the Drosophila outside in these cages, we had replication, and we tracked them over time. We tracked their changes in genotype frequency, we tracked their changes in phenotypic evolution. So these are populations that are meant to mimic something natural. We also had a second treatment, which I'll call non-seasonally evolving, or N. What we did here is we had these populations outdoor. They're producing eggs every 2 days. We're counting the number of eggs. We're discarding those eggs, and we're replacing those eggs with the eggs from the founding population. So that was a lot of work. Why would we do that? That sounds like a very strange treatment. The reason why we did that is because we wanted to compare the population dynamics of the seasonally evolving and the non-seasonally evolving replicates to investigate whether and how local adaptation influences population dynamics. So by doing this replacement, we're trying to prevent evolution from occurring. Sorry, I thought I almost said. So by doing the replacement, we're trying to prevent evolution from occurring. We're fixing the genotype frequencies by replacing the eggs every generation. We're not allowing or disallowing evolution across generations. So the question here was, how does or whether rapid evolution meaningfully influences the number of individuals and the persistence of population. So this E here is the seasonally evolving. We're not perturbing them in any way. And this N here is where we're counting the eggs and replacing them with eggs from the founder population. So we're allowing ecological change in this N. We're just not allowing changes in genotype frequencies across generations. We're stopping that from happening by replacing. What we saw was that if you stop rapid evolution from happening, all the cages, and this is mean census size, and this is from across 10 cages. If you stop that from happening, they all go extinct. All the Drosophila cages go extinct by about mid-September. So this is a little surprising, and it was a little hard to understand. So the other lines here, this black line is the daily maximum temperature. This gray line is daily minimum temperature. Drosophila melanogaster are from really warm climates. And we saw this big difference occur once the minimum temperature started to drop low around like 7-8 degrees, which is a little cold for Drosophila. But this seemed like a really strong effect to see differences. Is it possible that rapid evolution really had this meaningful of an effect on population dynamics? So we repeated this entire experiment in a subsequent year with another 10 replicates for each of these two treatments. And although there's definitely some year-to-year variation, we saw largely the same thing. So once conditions started to get cold, in this case, not even quite as cold, we saw a big divergence in the populations that were rapidly evolving and those where we were disallowing evolution by doing these replacements. So the second part of this question, the second part of this study was a question about whether rapid evolution alters the effects of rapid evolution on demography, on population size, feedback to shape natural selection. So again, this Lewontin quote about rapid evolution influencing the environment, in this case, the environment's density, does that feedback to drive additional rapid evolution? So the specific question in this case was, given that local adaptation led to increases in population size, we're predicting that an increased population size would feed back to drive evolution in traits associated with high density. So if there are true eco-evolutionary feedbacks, local adaptation leads to increases in mean fitness, increases in density. We saw that between the E and the N cages. And that higher density will feed back to just drive adaptation in response to high density. That's the prediction. To test that, we had one additional treatment, which I call no feedback or evolving density. I'll use ED as the shorthand here. So what we did in these cages was we counted the eggs every second day, and we reduced the density of these populations to match that of the populations where there was no evolution occurring. So this is getting like kind of deep in the weeds of evolutionary ecology, but the idea is we have a set of populations in which we're preventing evolution from occurring or replacing the eggs every generation. In that population, there's no effect of rapid evolution or on density. And so here we're allowing these to evolve, but we're not allowing the density to feed back to shape further evolution. So we're trying to decouple this eco-evolutionary. So stated perhaps more succinctly, this manipulation allows the ED populations to change in genotype frequency in response to natural selection, but removes any density-driven feedback from evolution onto population dynamics. So I'm knocking out this part of this potential eco-evolutionary feedback. So the seasonally evolving, or the E cages, should show evidence of local adaptation to increase density. They have this feedback present. I haven't knocked anything out. The no feedback treatment should not show evidence of evolution in response to high density. So what did we see? What we saw, this is an assay here where we're looking at larval development and high density between the E and the ED. So the ED is the no feedback, the knocked out with density. We see that this is August, by August, we see big differences in the ability of these flies to develop under high density conditions. E cages show a higher rate of larval development and a higher overall eclosion in high density. This looks like evidence of adaptation to high density conditions. But we see this consistent across the next time point. So this effect sticks around for a while. This seems like a pretty persistent effect, and it corresponds with when we saw the highest egg density. So it seems like we're seeing evidence here of an eco-evolutionary feedback mediated by density. Somewhat importantly, this wasn't a difference that we saw in larval development rate independent of density. So this is larval development rate in low density conditions. We see the same overall rate of larval development, or at least not a major difference between the E and ED cases. So something specific, we see evidence specifically of evolution to deal with high density conditions when you have the feedback. When you don't have the feedback, we see evidence that there's less of that. So local adaptation led to increased population size. That's the effect of evolution on the environment, population size of the environment, which led to adaptation to high density. So this is a demonstration that can be dynamic interaction and dynamic feedbacks between ecological and evolutionary change over short timescales. We also sequenced these populations and looked at differences between the E and the no feedback cages. So what was the genetic basis of this differentiation? And I put some genes on here that are some large effects, some of them are transcriptional regulators, some canonical Drosophila genes, but overall, the differentiation was relatively modest. There's a number of sites, a number of windows, some of them contain some interesting genes, but I'm not going to go through them too deeply. So the main takeaways from this part are that rapid adaptation and ecological time are occurring simultaneously. This quote from Lewontin, we're playing this out with an experiment in nature. And so populations adapt in response to continuous environmental shifts. We saw that previously. With adaptation has strong effects on population dynamics. Without rapid adaptation, populations went extinct a full 7 or 8 weeks before winter actually set off. And then finally, this adaptation feeds back through ecological mechanisms to shape further adaptation. So the work I was talking about, I did previously before I got to Washington State. I've only been to Washington State about a year, but a lot of that year was spent in getting this experimental site up and running. And so we now have an experiment running there, and I want to, since I spent so much time on it, I want to show this cool drone video that I took of the site that shows you this guy. This is the it's choppy. That's my postdoc Renee, and he's entering a cage. Each one of these cages often house 150 to 200,000 Drosophila, so you get large population sizes, which is good for evolution experiments. And we have plenty of room for expansion, so only limited by our ability to do so. Oh, that's not the end of the show. I have more. Okay, so what are we working on now in the lab? We're digging a bit more deeply into how aspects of genetic diversity influence the outcomes of adaptation. So how does the distribution, identity of genetic variation influence the pace and magnitude of rapid adaptation? A lot of the ways we're thinking about that is changing the genetic composition of the starting population, changing aspects like the presence or absence of inversions, the amount of genetic variation, whether we have putatively key variants, how much of those things actually shape the outcome of evolution. The other thing that I'm trying to work on, and this is more at the nascent stage, is we see in these Drosophila experiments this evidence of rapid adaptive tracking, that populations are adapting in response to momentary environmental change. Is there any generality of that? Are there other systems in which this question can be asked and it can be found whether this is specific to something in this particular multivoltine insect, or whether it's something that's happening in many different organisms out there in nature? I also want to plug the work of my postdoc, Renee Shah Mohamedloo, who's joined the lab and who's an evolutionary toxicologist. He's asking some of the very same questions, looking at rapid evolution within harmful algal blooms. And so Renee, the toxicologist, is a lot of expert in microcystins, and they produce microcystins, which are toxic to humans, toxic to aquatic life. And he's looking at the extent to which rapid evolution occurs within these harmful algal blooms using some genomic tools we're developing together. And so my team, in this case, this is Renee's team, is advancing by combining growth trials, modeling, and large-scale mesocosm experiments to track evolution in both microcystis and in things that feed on microcystis, to try to understand what role rapid evolution might play in something that's a little bit more ecologically important than Drosophila. In this case, harmful algal blooms that are a major ecological disturber. And so with that, I'll say thank you again to Mark and to everyone for listening, and say that come visit at WC Vancouver if you're interested. We'd love to have you come and visit and give a seminar if you're interested. And also that I'm hiring at every level. So looking for master's students, PhD students, technician, bioinformatician, postdoc. If you're interested in the work or know somebody is, please share. Thank you.
A lot of time for questions.
Francis.
I'll ask one. Thank you, but I was a tour de force. So my head's trying to catch up. But I was thinking about whether Lewontin is mostly right or mostly wrong. And what I mean by that is, we know that we'd like to have species have a prominent role in ecosystems, but oftentimes in ecosystem, only a really small handful has an imprint on ecosystems. And of that, I would imagine that a small percentage will be able to reap the benefit of any ecosystem modification exclusively, because they're going to be cheaters, right? But the other side is, does ecosystem slow to kind of, is that a barrier to rapid evolution? Because I think about phosphorus cycle in lakes, I think about soil organic matter pools or DOC pools in the ocean, like those are really slow pools that should just kind of hit the brakes on rapid evolution because it is not going to change for another 1,000 years.
Yeah, I mean, I think there's a lot of ways to answer that question. Is Lewinton mostly right or mostly wrong? I don't think we know yet. I think I could spend a long time trying to figure it out and still might not know. I would say that there are definitely stops in cases where you wouldn't expect evolution to be rapid. So one, and this is, people talk about this all the time in evolution, is that the environment is pretty constant. I think I would challenge that one. I think in many, many cases, I'm sure there are some in which things are constant, but I think that there are aspects of the environment, whether that be conspecific density, changes in interspecific interactions, changes in the biotic or abiotic environment of any kind, in which there probably are, of course, there are some that are stable, but a lot of them I think are changing relatively rapidly over time. The second part is the evolution part, the pace at which evolution would actually occur and how many organisms actually have a strong enough effect on the surrounding community or ecosystem to actually matter in their own selective landscape. I think if you think about it as evolution has to have an effect on the broader community or ecosystem functions, it's probably a smaller number of organisms. But if you think of the environment as also being conspecific density, I think it's a little bit, you could have that being more. And so I think it depends on where you draw the line between aspects of the environment.
Yeah, it's how you say it, E.
Yeah, if the environment is like ecosystem functions, then like, yeah, you're talking about like some bacteria or maybe some top-down control, but it's a little more limited. But if it's the E is also conspecific density, then that's probably almost everything, right? Molly?
Yeah, really nice talk. This is really cool stuff. So I have two questions, maybe three. First of all, how do you ever see any evidence of in the mesocosm contamination from outside flies?
So never any evidence. We worry about this a lot. So to the point where we drive metal like through the bottom to prevent any from coming underneath, and everything's kind of screwed in the bottom of the cage frames and the mesh. A lot of effort goes into that. How do we validate it? The best way that I've tried in the past is looking at genetic distance between neighboring cages with the idea that if there was flow in or out into neighboring cages, you'd see that neighboring cages are more similar over time or become more similar. Never seen any evidence of that. Keeps me up at night though, so I hope I never.
I feel like we can never even rule out even in the tinier lab experiments. So
yeah, I think in some ways it's easier here because if they were to get out, getting back into another cage would be so hard. I feel like their probability to get into another cage, you have to go, they're not very good at burrowing. So I mean, that sort of helps. But yeah, I've never seen it. It's never been enough to detect at a population level, but I can't say that it never, ever happens.
right, in which case I guess it wouldn't really matter to your bigger conclusions.
It matters when I try to do experiments where I manipulate the amount of genetic diversity, like just a few immigrants in a low diversity population, that would be bad. But I did an experiment and talk about it today where I started some cages with very low diversity and that diversity didn't go up. So I never saw an increase. There wasn't any new alleles showing up or there wasn't any new heterozygosity cropping up later in the experiment, which would have been a little scary. So I haven't ever seen, but yeah, for this stuff, it doesn't matter as much.
Okay, well then second question, since it sounds like you've messed around with different starting populations with different levels of genetic diversity, where do you come down on the idea of how general parallelism is, like the relationship to that generality to what you're starting with in your ancestral population? I feel like there's a little bit of debate about that in experimental evolution, because not everybody starts their experiments the same way.
Yeah.
It sounds like you and Mark would start with a single population and sample it multiple times and then see how all of those adapt, but others are doing it differently and finding less parallelism.
I think it matters a lot. And Mark and I were just talking about this like before seminar. It matters a lot in two ways. I think if you start with something that has large haplotypes, you're maybe a lot more likely to see parallelism, at least in this case. And that's what we've seen. So I've started some of these cages with inbred populations. So that means no diversity, totally inbred lines. You recombine them into a sort of a synthetic outbred population. So you reintroduce diversity by recombining the lines, and then you start the experiment. I've also done them with isofemale lines. So that means there's already variation within the line. You didn't purge the genetic diversity, you recombine those and you start them. This is an N of 1 because I've only done one of each type, but you get differences in what the architecture looks like. You see cleaner peaks when you start with inbred lines, is what I would say. So then the other part about how much does starting genetic diversity lead to differences in parallelism, very interested in doing that. I think my PhD student, Katie, might want to do that this summer, where you start with different amounts of genetic diversity and you see how much that influences the pace and magnitude of evolution, but also the amount of parallelism within independent populations. You might think that with lower diversity, maybe there's fewer paths, fewer genetic paths to adaptation, or maybe not. So I don't know, but I'm interested in that.
No. So you have these fluctuations. Sorry, great talk, awesome. But you have these fluctuations in populations, and we're kind of like now living under the like specter of climate change in like a more stochastic climate, more stochastic environment. Do you expect these rapid fluctuations and rapid ability to evolve to be like offer more resilience or more of a chance to go extinct? Because when you have lots of stuff varying really quickly, right, you can get those stochastic extinctions really easily.
I think if I was going to be bold about it, I'd say that I think that rapid evolution might be an important part of population persistence now. That there's this great paper called Eco-Evolutionary Dynamics, the underappreciated role that rapid evolution might play in population persistence and the way that biodiversity persists now. I think maybe that is true. Maybe if you were to remove evolution from every species today, or from some select number, that you might see extirpation in a lot more than you might think, at least multi-boltons. No, I don't think it's gonna be, I don't think that, so I should say that changes that we see in genotype, we don't see genotypes going to fixation very often. So we're not losing diversity due to like really strong selection that goes up, and then otherwise it couldn't fluctuate back down at an individual rule set. So very rarely, or almost never, we see fixation within our experiments. And that's true even of laboratory evolution experiments. You very rarely see selection being strong enough to actually like purge variants. You see it mostly changing the relative frequencies of different variants.
Sorry, it's a naive question because I'm in college. I don't know much evolution stuff, so I was like,
Bruce, how do you physically sample your agents?
Good question. We do it two different ways. We use this vacuum system that I modified to vacuum them out of the air. And I like that way because you're laughing, but this is a major innovation. I'll share this out with you, Mark, if you're interested. It is a major innovation in fly sampling because it takes a long time. So if you want to create a random sample, you need to sample all of the sub-habitats within your cage. That's non-trivial. If you just set a bottle in there like a trap, you're going to get only the flies that go in. The other way we've sampled is they only have a limited amount of media, so you can collect eggs and then use those eggs and rear them up. But it depends on what we're sampling for, I guess.
So do you actually try to go into the cage or?
Oh yeah. We're in there all the time, which some people don't like because, as I said, there's a lot of flies in the cage. So if you're not into swarms of Drosophila, you wouldn't like it. Oh yeah, sorry. Yeah. So there's someone in the cage in this. He walks into the cage.
How do you keep them from getting out when you walk in?
So you sort of shake the door so they get off the door, and then you zip it up and zip it behind you. Yeah.
We have a first question.
How did I decide to set them up at this scale? I would say that I contacted a manufacturer of mesh cages and asked what sizes they were able to make, and then kind of picked what was realistic. I mean, I don't have a real size limitation. They could be 10 times as big or potentially smaller. Realistically, my cutoff and the reason why it's 2 meters is because I needed to go inside of them. I don't want to be crouching down, so that's about a fit. That's the answer to that one. Well, let's see a picture. Smaller. Is there a population threshold population size? I'm sure there is a threshold population size if I had to imagine. And I like to do experiments about that where if you hold, so people talk in evolution so much about the role of NE, the amount of genetic diversity within a population as as shaping evolutionary outcomes? What about N? What about how many individuals you have? That dictates how many unique haplotypes, how many unique combinations of alleles you have. That's really important too. I'm sure that if you push it down far enough, you probably don't get adaptation. Also, sometimes populations start to decline and then they'll bounce back up. If you're close enough to zero, zero is an absolute number. You know, once you get far enough down, you're going to get extirpation. So I bet it matters a lot. There was another part of that question.
That's all.
Okay, cool.
So I have kind of a related question. So to me, an interesting aspect of this is thinking about the role of plasticity in all of this and how that might vary from system to system and levels of variation.
Yeah.
I guess the second part, so you have that egg swapping. Are you able to assess the role of plasticity using that data in any way?
Kinda. So we've tried this other ways too. So all the phenotypic evolution data I showed was after 2 generations of common garden variety because I want to measure the rate of evolution. But that's not the phenotype of the flies in the cages. The actual phenotype in the cages is the result of whatever genetic changes occurred, but then also all of the plastic changes that have occurred. And so sometimes you'll see really big differences. We've scored this sum where you measure phenotypes from flies collected directly from the cages, and you see change, let's say body sizes going down. But then you rear them in a common garden environment and you see evolution towards a larger body size. And so you can see that there are aspects of the environment in which it's driving plastic change, but evolution is acting sort of counter gradient or counter current to that. But we haven't, just gets to be a lot of phenotyping because if you're measuring the traits in all the cages, both from the cages themselves and then after 2 generations, it kind of pushes the upper limit of what's manageable for our lab. So we haven't included it regularly as part of our workflow. But were you thinking about something specific when you said the replacement? The fact that those replacement eggs are not going to be any plastic changes, they won't have those. Yeah. I think about that a lot too. So when we tried to sort of stop adaptation, it's really hard to do that. So we did these replacements, but these eggs were laid by mothers who were experiencing a static indoor environment. And so, you know, that would be a negative there because any sort of epigenetic changes that maybe aren't so important in flies, but are really important in other organisms wouldn't be there. The one thing that I think somewhat ameliorates that is that even in this no evolution cage, there actually still is evolution. It's just that every generation is acting on the standing crop of eggs. So it's like a single generation of evolution occurring over and over and over again. So it might be an underestimate, it might be an overestimate of the effect, but see what happens. Maybe.
I guess like you mentioned that you had tracked them like from fall to winter. Could they like survive through the winter and then bring them back to spring to see if they like do the same?
Yeah. This is the very obvious question that I'm very embarrassed to say is we don't know. So people have been studying Drosophila even in the seasonal evolution context in natural populations for like well over a decade now. And they're there in the spring. Spring is relative. You really don't see them at high density until like June or July. That sort of spring, and then they're there well into the fall. We know based on sequencing of populations over space and time that they are overwintering. There's isolation by distance, so it's not like they're recolonizing from Florida every year. But we've never been able to successfully have them overwinter in our cages. I think there's a much higher likelihood of that here than when I worked in Pennsylvania. The winter is less severe, so that's a big part of what I hope happens over this winter is that I can estimate, I can allow selection to occur over winter and see whether there is things like loss of genetic diversity that might be something that you wouldn't typically see under our normal selection period.
Really don't know that?
Well,
Crazy.
well, there's not a lot of Drosophila ecologists out there.
Wow.
So
Nobody cares what they do in the wild.
no, when you go to a Drosophila meeting, people are not talking about what they do in the wild.
Wow.
This would be like this is pretty far on the ecology side as far as Drosophila go, I would say.
Yeah.
But also it's not super easy to collect a Drosophila in the winter because how would you do that, right? Normally you might set traps or baits, but they might not move if it's cold. So you're like actively searching in the soil for a Drosophila, which sounds really hard.
Might just be the eggs.
It might be, but I think on the physiology, I think it's a little more likely it's the adults. They have this reproductive diapause they undergo. I don't know for sure, but if I had to guess, it's probably adults. But yeah, again, maybe some people know and I just don't, but I've asked a lot of folks and no one has any winter Drosophila collections.
There's one more question on Zoom.
Yes, generation time changes quite a bit with seasons. When it's hot outside, generation time's like 7 or 8 days. I have an experiment running now. This is like early fall, mid-fall. It's more like 18. And towards the end of the experiment, the eggs fail to develop at all. So below a certain critical thermal threshold, there's not enough degree days, there's not enough temperature to actually for development to occur. So yeah, it varies quite a bit over season.
Is lifespan the same? Are they like, do they live longer when it's hotter and shorter when it's colder?
Let me think about that. So people have tried to do lifespan in Drosophila. It's actually a little hard because you can't really follow individual flies so easily. So you try and measure it in different ways. I don't know whether it varies over time. It definitely lifespan's long enough over the course of these experiments where maybe there aren't original flies in the experiment, but you can imagine that there's a big time component, like the population itself acts as a reservoir of genetic diversity. Because flies that are 50 days old could still potentially reproduce, probably at a much lower rate, but contribute to the population. So if it goes like this, if it's a U-shaped environment, flies might not reproduce at all for 30 days and then come back, and their alleles could be increasing in frequency from the exact same mother, hypothetically.
Yeah, just a quick, so the population size thing. First of all, really nice talk. I'm curious about, I inferred from that you have like hundreds of thousands of flies swarming around in
Maybe like 200,000. Oh yeah, it's a lot.
So I'm curious how that might compare to actual natural effective population sizes for Drosophila out there. Like,
Any or N, like effective population. So any would depend on the experiment. I think sometimes where we've used a lot of isofemale ants to start, you're getting close to like a relatively realistic any for a specific area. N varies so much over space and time in Drosophila. You might go melanogaster, you might go to an orchard and you trap and you catch many, many insects and there's no melanogaster. You might go there and there's like millions and millions and millions of flies. So the magnitude of variation over space and time in the population size of melanogaster makes it kind of hard to answer that question. I think that the way that I think about these populations and the reason why I put my effort into doing these cages that have high population size is that it's getting us closer, at least within like an order of magnitude or something on N and NE of what populations may be like for multivoltine organisms, in terms of trying to understand evolutionary dynamics. Because if I was to do the same thing in a vial, it's only 50 flies, you're going to see a lot more effects of drift. NE's going to be lower, N's going to be lower. Yeah, I think it's getting closer, but I wouldn't ever say that it's the same.
Okay, well, thanks for coming, everyone.
Thanks, everybody.